Okay, let's unpack this. We often use these uh, broad terms like bad genes or, hereditary defects.
Right.
But those phrases, they really mask the stunning, almost microscopic precision of the errors that actually drive genetic disorders.
They absolutely do.
So today we're doing a deep dive into the very language of that error, the precise molecular architecture of genetic mutations.
Sounds good. It's a crucial area.
And when you look at the sheer scale of the problem, it really underscores why this is so fundamental to human health.
The numbers are quite stark, aren't they?
Yeah, current estimates suggest the lifetime frequency of genetic diseases is somewhere around 670 per 1,000 people. That's huge.
It is. And the earlier impact is even more dramatic.
Exactly. If we learn earlier, like in gestation, the data shows that a staggering 50% of all spontaneous early abortuses have some demonstrable chromosomal abnormality.
Half. It's not a footnote, as you say. It's central.
It's absolutely fundamental biology. So our mission today is to strip away those generalizations and really explore the molecular exactitude of what we call a permanent change in the DNA, the mutation itself, and trace exactly how that tiny error can cause these cascading systemic diseases.
And that specificity is absolutely key. The simplest definition, like you said, is a permanent change in the DNA structure.
Right.
But for understanding disease, the immediate implication really depends on where it happens in the body.
Okay.
So we first have to distinguish between two major locations. If a mutation occurs in the germ cells, that's the sperm or the egg.
The reproductive cell.
Exactly. Then it becomes part of the inherited genetic material. It gets transmitted to the next generation, and that's what leads to inherited diseases. That's the classic pathway,
That's the one we usually think of. So what's the alternative?
The alternative is when the mutation arises in somatic cells. That's basically any cell in the body that isn't involved in reproduction.
Okay.
Now, these mutations are not hereditary. They won't be passed down to children. But, and this is critical, they are incredibly important. They are essentially the engine behind virtually all cancers, allowing cells to escape normal growth controls. And they're also implicated in causing quite a few non-hereditary congenital malformations.
So understanding those two tracks, germline versus somatic, helps us place the single gene defects we're focusing on today.
Precisely. It gives us context. When physicians categorize human genetic disorders, they usually think in, let's say, three major buckets.
Okay, what are they?
First, you've got the disorders related to mutations in single genes. These are our focus today, the classic Mendelian disorders that usually have large, predictable effects.
Got it.
Second, there are chromosomal disorders. These involve really massive changes, structural alterations in chromosomes, or even having the wrong number, like an extra chromosome.
Like Down syndrome.
Exactly. And finally, the largest and arguably the most common category is complex multigenic disorders.
Right, things like heart disease or diabetes.
Perfect examples. These diseases don't usually arise from one single major error. Instead, it's the subtle, lifelong interplay between dozens, maybe hundreds, of common gene variants, we call them polymorphisms, and crucially, environmental factors.
So today we're drilling down into that first category. The single gene errors, where the mistake is identifiable, singular, and often, devastatingly powerful.
That's the plan. And the power of studying these single gene defects, as you hinted, is that they act like these molecular wrecking balls.
Yeah.
By seeing exactly how a system fails when just one piece is broken or missing, it actually illuminates the function of the entire pathway. It teaches us really profound lessons about just fundamental human physiology.
Okay, let's get granular then. Let's examine the sort of molecular physics of the error itself, starting with maybe the simplest but often incredibly impactful defect. The point mutation. This is literally, like you called it before, the single base switcheroo.
Yeah, the switcheroo. Point mutations are the most subtle type of genetic change, fundamentally. It's just one nucleotide base being swapped out for another.
Just one letter change in the code.
Exactly. But because the genetic code is read in these three-letter words, the codons, that single change can alter the amino acid that gets put into the protein chain. Or sometimes it can introduce a really catastrophic error.
So how do we categorize the results of that swap?
Well, functionally, we mainly talk about two types. The first is a missense mutation.
Missense.
This is where the base change alters the amino acid code, so a different amino acid gets put in, but the protein chain itself keeps getting built. It just incorporates the wrong building block at that one specific spot.
Right, but even within missense, there's a difference in severity, isn't there?
Oh, absolutely. There's a crucial nuance there. We divide them into conservative and non-conservative.
Okay.
A conservative missense mutation swaps one amino acid for another that's biochemically pretty similar, like maybe swapping leucine for isoleucine. Both are kind of greasy, hydrophobic amino acids.
So it might cause a subtle change, but maybe not break the whole protein.
Often, yeah. It might cause a little structural shift, but often results in minimal functional problems. It's like swapping a beam for one that's almost identical, as you said earlier.
But the non-conservative missense mutation, that sounds like the real danger.
That's absolutely right. The non-conservative switch replaces the normal amino acid with one that is structurally or chemically just incompatible, way different. And that can lead to massive functional consequences.
And the classic example here has to be sickle cell.
It's the textbook case, and for good reason. The normal DNA triplet in the gene for the beta-globin chain is CTC. That codes for glutamic acid.
Okay.
Now, glutamic acid is relatively large, it's polar, it's hydrophilic, it likes being around water.
Right.
The sickle cell mutation is a single base substitution. That CTC changes to CAC, and TAC codes for valine.
And valine is different.
Very different. Valine is small, nonpolar, hydrophobic, it hates water.
So just that one change, swapping a water-loving molecule for a water-hating one, what does that do systemically?
That tiny, tiny change at the molecular level forces the entire hemoglobin molecule to behave completely differently.
How so?
Under conditions of low oxygen, those hydrophobic valine residues, now sticking out on the surface, cause the hemoglobin molecules to literally stick together. They aggregate, they polymerize into these long, rigid fibers inside the red blood cell.
And that distorts the cell shape.
Precisely. It forces the red blood cell into that characteristic crescent or sickle shape. These sickled cells are fragile, they break easily, that's hemolysis.
Right. Anemia.
Yes, anemia. And they're rigid, so they clog up small blood vessels. That's vascular occlusion. It's just a stunning example of how one single molecular difference leads to this systemic pleiotropic disease with effects everywhere.
Incredible. Okay, so that's missense. What's the second main type of point mutation?
The second type is the nonsense mutation. And you can think of this as basically planting a premature stop sign right in the middle of the genetic sentence.
Okay, stop sign. How does that work?
So, the point mutation changes a normal codon 1 that codes for an amino acid into one of the three special stop codons. In mRNA, those are UAA, UAG, or UGA.
And the cell machinery recognizes those.
Instantly. As soon as the ribosome hits a stop codon, it halts translation. The protein synthesis just stops right there.
So you get a short, incomplete protein.
Exactly. If we look at another condition related to beta-globin, certain severe forms of anemia called beta-thalassemia are caused by nonsense mutations.
For example?
Well, if the codon for the amino acid glutamine, which is CAG, gets mutated to UAG, that's a stop codon, the protein synthesis machinery just stops dead.
And the result?
The result is a short, incomplete beta-globin peptide. It's almost certainly non-functional, and usually the cell's quality control systems recognize it as garbage and degrade it very quickly.
So the disease isn't caused by a bad protein, but by having basically no protein.
Precisely. A total lack of the necessary full-length protein product.
Okay, so we've covered swapping one base out. Now let's talk about changing the length of the code insertions and deletions, adding or removing bases.
Right. Small deletions or insertions, even just one or two bases, can fundamentally affect the coding sequence. And we categorize them based on how they mess with that triplet reading frame.
Okay.
First, you have what are called in-frame changes. This happens if the number of base pairs that are inserted or deleted is 3, or a multiple of 3, like 6 or 9.
So it adds or removes whole codons.
Exactly. The reading frame itself stays intact. The ribosome keeps reading triplets correctly downstream from the change, but the resulting protein will either gain or lose one or more amino acids at that spot.
It's like a flaw in the construction, maybe a missing brick or an extra one, but the rest of the blueprint is still readable downstream.
That's a great analogy. And the canonical example here, known worldwide, is the most common mutation causing cystic fibrosis, the delta 508 allele.
Delta F508, what does that mean?
Delta 500 and over 8 tellers represents a 3-base deletion. Those 3 specific bases code for the amino acid phenylalanine, F, at position 508 in the CFTR protein.
So it just deletes that one amino acid.
It removes the code for it entirely. The protein still gets synthesized, but missing that one crucial phenylalanine causes the protein to misfold really badly.
And the cell notices.
Oh, yes. The cell's quality control flags it for degradation in the endoplasmic reticulum before it can ever get to the cell surface where it needs to function as a chloride channel. So it's a defect in protein folding and trafficking, all caused by a clean, in-frame deletion of 3 bases.
Okay, so that's when the number is a multiple of 3. What happens when it's not a multiple of 3?
Now you're talking about a true molecular catastrophe,
Yeah.
the frame shift mutation.
Frame shift. That sounds bad.
It is. If the number of bases added or deleted is not a multiple of 3, say, you delete just 1 base or you insert 2 bases, the entire reading frame gets thrown off kilter from that point onward.
Right. It's like deleting a single letter from the beginning of a sentence. Every single word that follows becomes complete gibberish.
That's exactly what happens genetically. The ribosome keeps reading in triplets, but because the starting point shifted, all the downstream codons are now wrong.
So it starts adding completely incorrect amino acids.
Totally wrong amino acids. The genetic machinery incorporates a variable stretch of incorrect amino acids because the triplets are out of alignment. And this catastrophic misreading almost always, very quickly, generates a premature stop codon just by chance in the new garbled frame.
Leading to a short, useless protein again.
Severely truncated and almost always completely non-functional. We see devastating frame shift mutations causing many diseases. For example, a single base deletion in the ABO gene locus is what produces the non-functional O blood type allele.
Interesting.
And another key example is a 4-base insertion in the hexosaminidase A gene. That specific frame shift is a major cause of Tay-Sachs disease, particularly in Ashkenazi Jewish populations.
Wow. Okay, so these errors in the actual coding sequence, the exons, are clearly critical. But building on that idea of, molecular integrity, what happens if the error isn't in the instructions themselves, but maybe in the formatting or the labels on the blueprint?
Here's where it gets really interesting. That's a perfect way to put it. The complexity of gene regulation means mutations don't have to be inside the exons to be deadly.
Right.
Mutations outside the coding zone can be just as potent. They can essentially prevent the gene from ever being properly expressed or processed in the first place.
Okay, so where do these non-coding mutations typically occur? Let's look at maybe 2 key areas, the promoter and the splice junctions.
Exactly. Mutations in non-coding sequences like promoter regions or enhancer sequences, these are the DNA segments that control when and how much a gene is turned on.
The control switches.
Precisely. They work by binding specific proteins called transcription factors, which then recruit the machinery to start making an RNA copy of the gene.
So the switch is broken.
If the promoter sequence is mutated, those transcription factors might not be able to bind properly. The actual protein coding part of the gene, the exons, might be perfectly fine, pristine even. But if the on switch is broken, the gene is rendered functionally silent.
So you just don't make the RNA.
Transcription is either markedly reduced or sometimes completely absent. This leads to a quantitative deficit, not enough of the final protein product. And that's a key mechanism in certain types of thalassemia, for example.
Okay. And what about the errors at the sort of margins, the splice junctions?
Right. These are errors that mess up the post-transcriptional cleanup crew. After a gene is transcribed into RNA, you get this initial long molecule called pre-mRNA. This pre-mRNA contains both the coding bits, the exons, and these non-coding intervening sequences, the introns, which need to be removed.
Splicing.
Exactly. During RNA processing, or splicing, the introns must be precisely excised, and the exons stitched together perfectly at specific points called splice junctions. This creates the mature, functional messenger RNA, the mRNA, that's ready to be translated into protein.
So a mutation there messes up the cutting and pasting.
Perfectly put. A point mutation right at one of these critical splice junctions can prevent that clean cut. If the cellular machinery misses the correct splice site, the resulting mature mRNA might accidentally retain a chunk of an intron.
Oh, that sounds bad.
It is. Or it might skip over an entire exon. Either way, you often end up introducing an immediate frame shift or a premature stop codon into the message, ensuring that no functional gene product gets made from that transcript.
It just highlights the incredible fragility, doesn't it? The whole system needs such precision.
It really does.
But there is one final, really special category of mutation we need to touch on. It doesn't rely on substitutions or deletions, but on something more dynamic. The trinucleotide repeat anomaly.
Yes. This is a fascinating and relatively more recent discovery in genetics. The mutation itself is inherently unstable, which is quite unusual.
Unstable how?
It's characterized by the amplification, the expansion, of a sequence of 3 nucleotides repeated over and over again. And these repeats almost always involve guanine G and cytosine C, like CGG or CAG repeats.
And they expand.
Yes, the number of repeats can actually increase, particularly when the gene is passed down through generations, especially during gametogenesis, the formation of sperm and eggs.
So the mutation gets worse over time.
It can, yes. The degree of amplification, the sheer number of repeats, often increases from one generation to the next. This phenomenon is called genetic anticipation.
Anticipation.
Meaning the disease might appear at an earlier age or be more severe in successive generations because that repeat number keeps getting higher and higher. It's a dynamic process.
So what's the mechanism driving the disease, and what's the classic example?
The prime example, the one that really defined this category, is Fragile X syndrome, FXS.
Ah, yes, a major cause of intellectual disability.
It's the most common inherited cause, yes. Fragile X involves the amplification of a CGG repeat located in the regulatory region, specifically the promoter, of a gene called FMR1 on the X chromosome.
Okay, CGG. How many repeats are normal versus disease-causing?
In normal individuals, the average number of CGG repeats is around 29, maybe up to 50 or so.
Yeah.
But in individuals with Fragile X syndrome, that repeat count just explodes. It can range anywhere from 250 up to 4,000 tandem repeats.
4,000. That's massive. How does that cause the syndrome? Doesn't make a weird protein.
Interestingly, no. It doesn't necessarily alter the FMR1 protein sequence itself. Instead, that massive expansion of CGG repeats in the promoter region leads to the silencing of the entire gene.
It shuts it down. How?
That huge cluster of CGG repeats triggers excessive methylation, the addition of chemical tags to the surrounding DNA. This hypermethylation effectively condenses the chromatin structure, packing it so tightly that the transcription machinery simply can't access the FMR1 gene to turn it on.
So no FMR1 protein is made.
Essentially, yes. The resulting lack of the FMR1 protein, which we now know is crucial for normal synaptic development and function in the brain, causes the intellectual disability and other features of the syndrome. It's a fascinating case of an error of over-repetition leading to a complete transcriptional shutdown.
Okay, so we've journeyed from the single molecular letter change, the point mutation, all the way up to these massive dynamic repeat expansions. Now let's zoom out a bit. The final result of any of these Mendelian disorders is ultimately a defect in the gene product, right?
Fundamentally, yes. It boils down to one of two things. Either an abnormal, dysfunctional protein is produced, or the body simply produces a drastically reduced amount, sometimes none, of the necessary protein.
And before we dive into specific examples of diseases tied to protein types, let's quickly nail down two concepts that are really critical for understanding the often messy systemic reality of these single gene diseases. The first is pleiotropism.
Pleiotropism, yes, very important. Pleiotropism describes the situation where a single mutant gene somehow manages to produce many, often seemingly unrelated, end effects across multiple different organ systems.
One gene, many problems.
Exactly. The initial error is singular, located in one gene, but the ripple effects are systemic, touching different tissues and pathways.
And sickle cell is the perfect example again.
It's our cornerstone example for pleiotropism. That single point mutation in the beta-globin gene leads to abnormal hemoglobin S, HbS.
Right.
This, in turn, causes the red cell distortion, leading to hemolysis, anemia. It causes vascular occlusion, those log jams in small vessels. And those vascular problems then cascade outwards, causing damage like splenic fibrosis, pulmonary hypertension, bone pain, increased risk of stroke, and widespread organ infarcts. It's one molecular change causing dozens of linked clinical manifestations throughout the body.
And this isn't just for structural proteins, right? We see pleiotropism in metabolic disorders too, say like phenylketonuria, PKU.
Absolutely. PKU is caused by a defect in the enzyme phenylalanine hydroxylase. That single enzyme defect prevents the normal breakdown of the amino acid phenylalanine. The buildup of phenylalanine and its abnormal byproducts doesn't just affect one thing.
What does it affect?
It causes severe neurological damage, leading to intellectual disability if untreated, but it also affects the dermatological system, causing eczema. And it causes hypopigmentation, lighter skin, and hair because the downstream pathway to produce melanin pigment is blocked.
So again, one enzyme error, multiple system effects. Brain, skin, pigment.
Exactly. It shows how pleiotropism applies whether the primary defect is in a structural protein like globin or an enzyme like phenylalanine hydroxylase.
Okay, and the concept that runs sort of opposite to that is genetic heterogeneity. What's that about?
Genetic heterogeneity is, in a way, the flip side. It describes the phenomenon where mutations at several entirely different genetic loci, different genes, can all end up producing the exact same clinical trait or disease.
So same symptom, different genetic cause.
Precisely. If you walk into a clinic and see, for instance, a child with profound congenital deafness, clinically, it looks like one condition.
Right?
But genetically, that deafness could be caused by mutations in any one of literally dozens of different genes. Genes involved in inner ear hair cell function, ion channels, structural proteins in the cochlea, protein trafficking, many possibilities.
Wow, dozens.
Yes, and recognizing this heterogeneity is absolutely essential in modern medicine. It means that for effective genetic counseling, or especially for developing targeted therapies like gene therapy, you need to know the specific mutated gene causing the deafness in that individual patient, not just the clinical diagnosis of deafness.
That makes total sense. Okay, let's transition now to thinking about how the type of protein being damaged often dictates the inheritance pattern.
Yeah.
We often see that defects in structural proteins, things like collagen or cytoskeletal elements, tend to result in autosomal dominant disorders. Why is that? Why does having just one bad copy of a structural gene cause disease, unlike most enzyme deficiencies?
This is a really important distinction, and it often comes down to something called the dominant negative effect.
Dominant negative. Okay, break that down.
Think about how structural proteins usually function. They rarely work alone as single molecules or monomers. Instead, they assemble into larger complexes, maybe dimers, trimers, or even massive scaffolds and fibers.
Like building blocks forming a structure.
Exactly. Let's take collagen, the main protein in connective tissue. Functional collagen exists as a triple helix, a trimer made of 3 individual collagen chains wound together.
Okay, 3 chains.
Now imagine an individual who is heterozygous for a mutation in one of the collagen genes. They have 1 normal allele and 1 mutant allele. So their cells produce roughly 50% normal collagen chains and 50% defective mutant chains.
Right. So why doesn't the 50% normal stuff just do the job?
Because of the assembly process. When the cell tries to assemble the triple helix, those mutant chains get incorporated randomly along with the normal chains. The problem is, even incorporating one defective chain into the trimer can destabilize the entire structure. It corrupts the whole complex.
Ah, so the bad apple spoils the bunch. The defective piece doesn't just fail to help. It actively sabotages the good pieces.
That's the core idea of dominant negative. The mutant protein actively impairs the function of the protein produced by the normal allele. In the case of collagen, even though half the individual chains are normal, the vast majority of the assembled trimers end up being structurally unstable and dysfunctional.
So the body doesn't just have 50% function, it has much less.
Much less. You end up with a marked overall functional deficiency of collagen far greater than 50%. This results in conditions like some severe forms of osteogenesis imperfecta, brittle bone disease. It's fundamentally different from simple haploinsufficiency, where just losing 50% of the product causes the problem. Here, the mutant product is actively interfering.
That concept, the dominant negative effect, seems like the perfect lens for looking at Marfan syndrome. That's the classic connective tissue disorder, right?
Absolutely. Marfan syndrome is caused by an inherited defect in the FBN1 gene. This gene codes for fibrillin-1.
Fibrillin-1, what does that do?
Fibrillin-1 is a large glycoprotein found in the extracellular matrix, the space between cells. It's essential for forming complex structures called microfibrils.
Microfibrils.
These microfibrils act like a scaffold. They provide the structural framework upon which elastic fibers are laid down, particularly in tissues that need to stretch and recoil, like the walls of the aorta, ligaments, and the ciliary zonules that hold the lens in the eye.
So defects in the scaffold explain the symptoms.
Largely, yes. The loss of structural support in these microfibril-rich tissues leads directly to the characteristic clinical manifestations. You see the skeletal abnormalities, long, slender limbs, loose joints, hypermobility, chest deformities like pectus excavatum. You see the eye problem, ectopia lentis, where the lens is displaced because the zonules are weak. And most critically, you see weakness in the wall of the aorta, the main artery leaving the heart. This leads to progressive dilation, aneurysm, and the life-threatening risk of aortic dissection or tearing.
But isn't there a more modern insight into Marfan pathogenesis, something beyond just the structural failure?
Yes, absolutely. This is where the story gets even more interesting and connects fibrillin-1 directly to cell signaling pathways.
How so?
It turns out that normal fibrillin-1 plays a vital regulatory role. It binds to and sequesters a potent signaling molecule called transforming growth factor beta, or TGF-beta-globin.
Sequesters, like it locks it up.
Exactly. It keeps TGF-beta inactive, stored away in the extracellular matrix, controlling its availability to signal to cells.
Okay, so what happens when fibrillin 1 is defective in Marfan syndrome?
When fibrillin 1 is defective or present in reduced amounts due to the FBN1 mutation, that sequestration mechanism fails. TGF-beta isn't properly locked down.
So it gets released inappropriately.
Yes. This leads to excessive and abnormal activation of TGF-beta signaling in tissues like the aortic wall. So instead of just having a weak scaffold, the tissue is now also being subjected to inappropriate signals.
And what does that excessive TGF-beta signal do?
It's actually detrimental. It promotes inflammation, it increases the production of enzymes that degrade the extracellular matrix itself, and it adversely affects the development and maintenance of the smooth muscle cells within the aortic wall.
So it's not just a passive structure falling apart. It's almost like an active inflammatory self-destruction driven by this rogue signaling pathway.
That's a perfect summary. And this deeper molecular understanding that excessive TGF-beta activity is a key driver has revolutionized potential therapies.
How has it led to treatments?
Because if the downstream problem is too much TGF-beta activity, then maybe we can block that activity. Clinical trials started looking at existing drugs, specifically a class called angiotensin receptor 2 blockers, or ARBs drugs like losartan, which were originally developed to treat high blood pressure.
And they affect TGF-beta.
Yes, it turns out that ARBs also have the beneficial side effect of inhibiting that detrimental TGF-beta signaling cascade.
Really?
Yes. Studies in both Marfan patients and animal models have shown that ARBs can significantly slow down the rate of aortic root dilation. It offers a rational, mechanism-based, non-surgical approach to mitigate the most dangerous aspect of the disease. It's a fantastic example of molecular insight leading to therapeutic innovation.
That's truly remarkable. Okay, let's pivot now pretty sharply. Let's move from structural proteins to mutations affecting enzymes. This covers the vast majority of those classic inborn errors of metabolism. And these conditions are overwhelmingly autosomal recessive. Why the difference? Why does the margin of safety usually work so well for enzymes?
Right. The margin of safety is a really fundamental principle in metabolic genetics. Think about a person who is heterozygous for an enzyme deficiency. They carry one normal allele and one mutated allele for that specific enzyme.
So they make some normal enzyme.
Typically, yes. They usually synthesize roughly 50% of the normal amount of that enzyme.
And 50% is usually enough.
For almost all metabolic pathways, yes. 50% enzyme activity is perfectly sufficient to prevent the toxic buildup of precursors upstream of the block, or to avoid a critical deficiency of the end product downstream. We seem to have evolved with a massive reserve capacity in most of our metabolic pathways.
So you need to lose function from both copies of the gene.
Exactly. It generally requires having two mutated alleles either being homozygous for the same mutation or compound heterozygous for two different mutations in the same gene to drop that total enzyme activity below the critical threshold, maybe 10% or 20% of normal, where clinical disease starts to manifest.
Are there any exceptions? Do we ever see dominant enzyme deficiencies where losing just 50% does cause a problem?
They're very rare, but they do exist. It might happen if the enzyme controls a really crucial rate-limiting step in a pathway where the product level is extremely sensitive, or sometimes, though less common for enzymes, if the enzyme itself forms part of a complex where a mutant subunit could exert a dominant negative effect, similar to the structural proteins. But as a general rule, it holds remarkably true. If the primary problem is a metabolic block due to an enzyme deficiency, the disease inheritance pattern is almost always recessive.
So when that block does occur in someone with recessive disorder, they have two bad copies. We generally see three major ways the disease can actually happen, three pathogenic pathways.
That's right. Three main consequences flow from that enzyme defect. The first, and probably the most common mechanism, is the accumulation of the substrate that the enzyme was supposed to act on, or the buildup of toxic intermediates from alternative pathways.
So stuff piles up before the block.
Exactly. If the enzyme is supposed to break down molecule A into molecule B and the enzyme is missing, molecule A builds up. If molecule A itself, or some byproduct formed from it when it accumulates, is toxic in high concentrations, it causes tissue injury.
Can you give an example?
Galactosemia is a classic example. Here, infants lack the enzyme needed to metabolize galactose, a sugar found in milk. Galactose and its toxic byproducts, like galactitol, accumulate, causing severe damage to the liver, kidneys, and brain.
Okay, accumulation is number one. What's the second consequence?
The second is much more straightforward. A decreased amount of the end product. The enzyme was necessary to complete a synthesis pathway, and if it fails, the necessary final molecule is simply missing or deficient.
Right.
The classic example here is albinism. In the most common form, there's a defect in the enzyme tyrosinase. Tyrosinase is essential for the first steps in synthesizing melanin pigment from the amino acid tyrosine.
So no tyrosinase, no melanin.
No functional tyrosinase means no melanin biosynthesis. This leads directly to the clinical phenotype, the absence of normal pigmentation in the skin, hair, and eyes. The problem is the lack of the final product.
Makes sense. And the third pathway, you said it was a bit more subtle.
Yes, the third consequence is the failure to inactivate a normally produced but potentially damaging substrate.
So the enzyme's job isn't to build something or break down waste, but to put the brakes on something else.
Precisely. This highlights the importance of enzymes that act as inhibitors or neutralizers. Consider alpha-DOR1 antitrypsin deficiency. Alpha-1 antitrypsin, A1AT, isn't involved in making a product in the usual sense. It's a protein, specifically a protease inhibitor, whose main job is to circulate and neutralize neutrophil elastase.
Or neutrophil elastase.
That's a powerful, destructive enzyme released by neutrophils, a type of white blood cell, particularly in the lungs during inflammation or infection. Normally, A1AT keeps elastase in check.
So what happens in the deficiency?
When a person is severely deficient in A1AT due to mutations in the SERPINA1 gene, that neutrophil elastase runs rampant, unchecked, especially during periods of inflammation like smoking or lung infections. It relentlessly attacks and destroys the elastin fibers that provide structure and elasticity to the lung alveoli. Leading ultimately to severe pulmonary emphysema, a chronic lung disease characterized by the breakdown of lung tissue. The disease results from the failure to inactivate a damaging enzyme.
This idea of, systematic failure in breakdown and quality control leads us perfectly into a really crucial subcategory of enzyme disorders, the lysosomal storage diseases. These seem central to a lot of modern molecular research, especially with links to neurodegeneration.
Absolutely. Lysosomal storage diseases, LSDs, are a prime example of enzyme deficiencies causing problems. And yes, their connection to broader neurodegenerative processes is a major focus now.
So remind us, what is the lysosome's job?
The lysosome is essentially the cell's recycling center. It's an organelle filled with dozens of different powerful hydrolytic enzymes, acid hydrolases, that are designed to break down all sorts of complex macromolecules, proteins, lipids, carbohydrates, nucleic acids into their basic building blocks for reuse.
A cellular garbage disposal and recycling plant.
That's a great way to think of it. But for this system to work, the cell needs an incredibly precise delivery system to get those powerful enzymes into the lysosome and keep them contained there.
Right, you don't want those enzymes leaking out.
Yeah.
How does the cell target them?
It uses a special molecular address label. As these lysosomal enzymes are being synthesized and processed through the Golgi apparatus, they get tagged with a specific sugar modification. Mannose 6-phosphate.
Mannose 6-phosphate. That's the tag.
That's the key tag. There are receptors in the Golgi, the mannose 6-phosphate receptors, that recognize this tag. They bind to the tagged enzymes and actively shuttle them into vesicles that eventually fuse with and deliver their contents to the lysosomes.
What happens if that tagging process fails?
If the enzymes aren't properly tagged with mannose 6-phosphate, the receptors don't recognize them. Instead of being sent to the lysosome, these enzymes get mistakenly secreted out of the cell. The result is a functional deficiency of multiple lysosomal enzymes inside the lysosome, even though the enzymes themselves might be perfectly functional. This causes a rare but severe LSD called I-cell disease.
Okay, but in most LSDs, it's a single enzyme that's defective, right?
Right.
So what happens then?
Right. In the more common LSDs, the problem is usually a mutation in the gene for one specific lysosomal enzyme. The targeting system works fine, but the enzyme itself is missing or non-functional.
So the stuff it was supposed to break down just piles up.
Exactly. That leads to the primary accumulation. The specific substrate, the macromolecule that the missing enzyme was meant to digest, remains intact and undigested. Because these substrates are often complex lipids or carbohydrates, they are typically insoluble and they build up progressively within the lysosomes.
And the lysosomes swell up.
Yes, the lysosomes become engorged with this undigested material, creating these characteristic swollen storage vacuoles that eventually disrupt normal cell function and architecture. This is the classic pathology you see in disorders like Tay-Sachs disease, where GM2 gangliosides accumulate massively in the lysosomes of neurons.
But you mentioned this modern critical link. How does this lysosomal failure connect to much more common diseases of aging, like Parkinson's or Alzheimer's?
This is where the field has really advanced. We now understand there's a fundamental, intimate, functional connection between the lysosomes and the cell's main waste clearance pathway, a process called autophagy.
Autophagy, self-eating.
Right. Autophagy is how the cell gets rid of old, damaged components like worn-out mitochondria or misfolded protein aggregates. It engulfs them in a membrane, forming an autophagosome, which then fuses with the lysosome. The lysosome's enzymes then degrade the contents.
So the lysosome is the final destination for autophagy.
It's the essential final step. Now, what happens if the lysosome itself is already clogged up or dysfunctional, as in a lysosomal storage disease?
It can't handle the incoming traffic from autophagy?
Exactly. Defective lysosomal function significantly impairs the entire autophagy process. The cell's main garbage disposal system gets backed up because the final processing unit is overloaded or broken.
So the trash piles up elsewhere.
Precisely. This failure in autophagy leads to what we call secondary accumulation. You get a massive buildup of things that should have been cleared out. Dysfunctional mitochondria, which spew out damaging reactive oxygen species, and the accumulation of toxic, aggregation-prone polypeptides. Proteins like alpha-synuclein, which is central to Parkinson's disease, and the Huntington protein involved in Huntington's disease. These proteins normally get cleared by autophagy.
So a primary lysosomal defect can indirectly cause the buildup of proteins linked to other neurodegenerative diseases.
That's the critical insight. It provides a strong molecular mechanism linking inherited lysosomal defects to the pathogenesis of much more common, often sporadic, neurodegenerative disorders of aging. It suggests that even subtle declines in lysosomal function with age could contribute to these conditions.
And we see this principle really starkly illustrated in Gaucher disease, right?
Yeah.
You said it's the most common LSD.
Yes, Gaucher disease provides perhaps the clearest example of this link currently. It results from a deficiency of the enzyme glucocerebrosidase, GBA. This leads to the accumulation of its substrate, glucocerebroside, primarily within cells of the macrophage lineage phagocytic cells. These lipid-laden macrophages are called Gaucher cells, and their accumulation causes symptoms like enlargement of the liver and spleen, bone problems, and sometimes neurological issues.
But what's the Parkinson's connection?
The compelling modern finding is that individuals with Gaucher disease, even heterozygote carriers with just one mutant GBA allele, have a significantly increased risk, perhaps 5 to 20-fold higher, of developing Parkinson's disease compared to the general population.
20-fold. That's huge. Why?
The evidence strongly suggests it's due to that impaired lysosomal function caused by the GBA deficiency. Even partial deficiency seems to hinder the effective autophagic clearance of alpha-synuclein. This leads to alpha-synuclein aggregation and the formation of Lewy bodies, the pathological hallmark of Parkinson's disease. It's a direct molecular link between an LSD gene and Parkinson's risk.
Fascinating. Okay, let's shift gears again. Moving from enzyme defects, let's consider problems with the machinery that moves things in and out of cells or allows cells to receive signals receptor and transport system defects. These often seem to follow an autosomal dominant pattern, like Morphan. Familial hypercholesterolemia, FH, is maybe the best example here.
FH is a perfect molecular demonstration, and yes, it's typically autosomal dominant. It's also a major public health concern because heterozygous FH affects roughly 1 in 200 people.
1 in 200? That's quite common. What's the core problem in FH?
The fundamental issue is a defect in the body's system for clearing cholesterol-rich low-density lipoprotein, or LDL, from the bloodstream. About 70% of LDL clearance normally happens in the liver, mediated by the LDL receptor.
So if the receptor system is broken.
If that receptor system is compromised, LDL cholesterol can't be efficiently removed from the plasma. It builds up to chronically high levels, leading rapidly to premature atherosclerosis, plaque buildup in arteries, and a greatly increased risk of heart attacks, often at a young age.
And molecularly, FH isn't caused by just one gene defect, is it?
That's right. The molecular diagnosis can be complex because clinically, FH can arise from mutations in at least 3 distinct genes, all involved in that LDL clearance pathway.
What are the main culprits?
The vast majority of cases, maybe 80–85%, are caused by loss-of-function mutations in the gene for the LDL receptor, LDLR itself. These mutations can prevent the receptor from being made, block it from getting to the cell surface, stop it from binding LDL, or prevent it from being internalized. Hundreds of different LDLR mutations are known.
Okay, so problems with the receptor itself. What else?
The next most common cause involves mutations in the gene for apolipoprotein B100, ApoB. ApoB is the protein component on the LDL particle that actually binds to the LDL receptor.
So it's the key that fits into the receptor lock.
Exactly. If you have a mutation in ApoB that changes the shape of that binding region, the LDL particle, the key, can effectively engage with a perfectly normal LDL receptor, the lock. The result is the same, impaired LDL clearance.
Makes sense. And the third gene, you mentioned it was the one that really changed drug development.
Yes, the third, and perhaps most fascinating from a therapeutic standpoint, involves activating mutations in a gene called PCSK9.
PCSK9, what does that do normally?
PCSK9 is an enzyme, a protease, that plays a crucial role in regulating the lifespan or turnover of the LDL receptor protein on the surface of liver cells.
How does it regulate it?
When PCSK9 protein binds to an LDL receptor, it essentially acts as a molecular executioner. It targets that receptor for degradation within the cell's lysosomes, preventing it from being recycled back to the cell surface to pick up more LDL.
So high PCSK9 activity means fewer LDL receptors on the cell surface.
Precisely. More PCSK9 means faster receptor turnover and degradation, fewer receptors available at any given time, and therefore higher plasma LDL levels. Activating or gain-of-function mutations in PCSK9 lead to FH by causing excessive degradation of LDL receptors.
Okay, but how did understanding PCSK9 lead to a revolutionary drug? You said it came from the opposite kind of mutation.
Exactly. This is a phenomenal story of genetic detective work leading directly to rational drug design. Researchers started noticing certain individuals and even whole families who had unusually low LDL cholesterol levels and seemed naturally protected from cardiovascular disease.
Lucky them.
Indeed. When scientists investigated their genetics, they discovered that these protected individuals carried rare loss-of-function variants in the PCSK9 gene.
So the PCSK9 didn't work well.
Right. These naturally occurring mutations resulted in little or no functional PCSK9 protein being produced. Without PCSK9 around to target them for destruction, their LDL receptors were degraded much more slowly. They persisted longer on the liver cell surface, clearing LDL from the blood hyper-efficiently.
So the natural experiment showed the way.
It provided the perfect therapeutic blueprint. If naturally losing PCSK9 function lowers LDL and protects against heart disease, then perhaps we can achieve the same effect pharmacologically by inhibiting PCSK9 activity.
And that led to the drugs.
Directly. This insight spurred the rapid development of highly specific PCSK9 inhibitor drugs, typically monoclonal antibodies that bind to and neutralize circulating PCSK9 protein. By blocking PCSK9, these drugs dramatically increase the number of LDL receptors expressed on the hepatocyte surface, leading to unprecedented reductions in LDL cholesterol levels far beyond what statins alone can achieve. It's precision medicine at its absolute best.
Truly amazing. Okay, let's shift our perspective slightly now, thinking about how an individual's specific genetic makeup influences their reaction to external things, particularly drugs. This is the field of pharmacogenetics, right?
Yes, pharmacogenetics or pharmacogenomics is really defining the path toward more personalized medicine. It's the study of how genetic variations influence individual responses to drugs, both in terms of efficacy and adverse reactions.
So it's about predicting how someone will react before they even take the medication.
That's the ultimate goal. A key aspect of pharmacogenetics involves identifying genetically determined enzyme deficiencies or variations that might be completely silent, causing no disease on their own, but are unmasked only upon exposure to a specific drug.
So the gene variant is like a hidden risk factor.
Exactly. The individual might be perfectly healthy under normal circumstances, but carries a genetic susceptibility that only becomes apparent when challenged by a particular chemical environment, like a medication.
What's the classic example of this?
The quintessential example, taught for decades, remains the deficiency of the enzyme glucose-6-phosphate dehydrogenase, or G6PD.
G6PD. What does that enzyme do?
G6PD plays a vital role in protecting red blood cells from damage caused by oxidative stress. It helps maintain levels of NADPH, which is needed to regenerate glutathione, a key antioxidant.
Okay, and the deficiency?
G6PD deficiency is actually quite common in certain populations, particularly those with ancestry from malaria-endemic regions, as it offers some protection against malaria. Crucially, under normal conditions, having G6PD deficiency usually causes no symptoms whatsoever. But if an individual with G6PD deficiency is exposed to certain triggers that increase oxidative stress in red blood cells, like specific drugs, the classic example being the anti-malarial drug primaquine, certain infections, or even eating fava beans, favism, their red blood cells can't cope.
Why not?
Because they lack sufficient G6PD activity to neutralize the oxidative onslaught. The enzyme deficiency is unmasked, leading to the rapid destruction of red blood cells, a severe, potentially fatal episode of hemolytic anemia.
So a drug that's safe for most people could be dangerous for someone with this specific genetic variant.
Precisely, and this principle extends far beyond G6PD. We are rapidly accumulating a huge amount of knowledge about common polymorphisms, these normal variations in genes that encode drug-metabolizing enzymes.
Like the liver enzymes that break down drugs.
Especially the cytochrome P450 family of enzymes, which are responsible for metabolizing a vast number of common medications. Variations in CYP genes, like CYP2D6 or CYP2C19, can dramatically affect how quickly or slowly an individual metabolizes drugs like antidepressants, antipsychotics, painkillers like codeine, or blood thinners like warfarin.
So the standard dose might be toxic for a slow metabolizer, or ineffective for a rapid metabolizer.
Exactly. We also see important variations in genes encoding drug transporters or the receptors that drugs target. Leveraging this knowledge, potentially genotyping a patient before prescribing certain drugs, allows physicians to start tailoring drug choices and dosages based on the individual's genetic profile.
Maximizing effectiveness while minimizing risk.
That's the promise of pharmacogenetics and personalized medicine. It's moving away from a one-size-fits-all approach.
Right. Okay, to kind of wrap up our deep dive into the sheer molecular complexity of these genetic errors, let's briefly touch on how all this incredibly detailed knowledge has actually led to advanced therapeutic strategies, especially for some of those devastating conditions we discussed earlier, like the lysosomal storage diseases.
Yes, the progress in treating LSDs is a direct result of understanding the underlying molecular defect, knowing precisely what the cell is missing or why a particular protein isn't working correctly. This has given rise to really 3 core therapeutic strategies.
Okay, what's the first one?
The first, and perhaps the most obvious approach conceptually, is enzyme replacement therapy, or ERT.
Just give the missing enzyme back.
Essentially, yes. The deficient lysosomal enzyme is manufactured using recombinant DNA technology, purified, and then infused intravenously into the patient on a regular basis, often every couple of weeks for life.
And does it work?
For many LSDs that primarily affect non-neurological organs like Gaucher disease, type 1 Fabry disease, Pompe disease, ERT has been truly transformative. It can significantly improve organ function, reduce storage burden, and enhance quality of life. Its main limitation is that these large enzyme molecules generally don't cross the blood-brain barrier effectively, so ERT isn't very useful for LSDs with major neurological involvement.
Okay, ERT is strategy 1. What's number 2?
The second strategy is called substrate reduction therapy, or SRT.
Substrate reduction, so reducing the stuff that builds up.
Exactly. The logic here is beautifully simple, almost counterintuitive. If the enzyme, say enzyme C in the pathway ABCD, is deficient, causing substrate C to build up, maybe we can partially inhibit an earlier enzyme in the pathway, say the one making C from B.
Why would you do that?
The goal is to reduce the overall flow of material into the defective step. If you can decrease the rate at which substrate C is synthesized, then perhaps the small amount of residual enzyme C activity that the patient might still have becomes sufficient to handle the reduced load, preventing toxic accumulation.
Lessen the burden on the broken part of the assembly line.
Precisely. SRT drugs are typically small molecules that can be taken orally and can often cross the blood-brain barrier. They are approved for certain LSDs like Gaucher disease type 1 and Niemann-Pick disease type C.
Interesting. Okay, and the third strategy, you mentioned it earlier as being particularly innovative, molecular chaperone therapy.
Yes, chaperone therapy is perhaps the most elegant manifestation of applying deep molecular insight. It addresses a specific situation found in many genetic diseases, including some LSDs.
What's the situation?
In many cases, the mutation doesn't completely destroy the enzyme's ability to function. Instead, it makes the enzyme protein structurally unstable. It causes it to misfold slightly as it's being made in the endoplasmic reticulum, ER.
And the cell detects this misfolding.
Oh, yes. The ER has very stringent quality control systems. It recognizes these subtly misfolded proteins as defective and targets them for premature degradation, usually through a process called ER-associated degradation, ERD. So the enzyme gets destroyed before it ever has a chance to reach the lysosome, even if it might have retained some activity.
So the problem isn't inactivity, but instability leading to destruction.
Correct. Molecularly chaperone drugs are designed to combat this. They are usually small chemical compounds, often specific inhibitors or analogs of the enzyme substrate, that can bind to the mutant misfolded enzyme within the ER.
And what does binding do?
This binding acts like a temporary chemical splint or scaffold. It helps stabilize the protein's correct three-dimensional conformation, essentially masking the subtle misfolding defect.
Allowing it to escape the quality control.
Exactly. By stabilizing the structure, the chaperone allows the mutant enzyme to pass through the ER quality control checkpoints, successfully traffic through the Golgi, get properly targeted, and eventually reach the lysosome. Once in the lysosome, the chaperone might dissociate, leaving behind a partially functional enzyme that can help clear the stored substrate.
That's amazing, like a therapeutic rescue mission at the protein level.
It really is. It's being used or tested for certain forms of Fabry disease, Pompe disease, and even some specific mutations causing Tay-Sachs disease. It requires incredibly detailed knowledge of the specific mutation and its effect on protein folding, making it a prime example of precision therapy.
Okay, this has been a phenomenal journey through the molecular landscape of air.
So what does this all mean, really? We started by defining that fundamental concept, the mutation, as a permanent change in DNA. And we've meticulously traced its potential effects, moving all the way from a single base substitution, the point mutation, through the chaos of frame shifts, right up to that dynamic, unstable amplification of trinucleotide repeats. And we established that key principle. The clinical outcome, particularly the inheritance pattern, often hinges crucially on the type of protein affected. Structural proteins frequently lead to dominant negative effects, causing disease even with one good copy.
While enzymes usually rely on the recessive margin of safety, needing 2 hits to cause disease.
Exactly. This whole journey confirms that the language of genetic error is incredibly precise, but also incredibly varied. And the insights we've gained from dissecting these specific failures, understanding Marfan or Tay-Sachs or FH at the molecular level, haven't just advanced our basic knowledge.
No, they've directly led to new therapies.
They've provided the exact molecular targets needed to revolutionize medicine. Think about designing PCSK9 inhibitors based on a natural protective mutation, or using chemical chaperones to physically rescue misfolded enzymes. It's a direct line from basic science discovery to clinical application.
And I want to leave you, our listener, with this final, maybe slightly provocative thought. We focus today mostly on mutations with massive, clear-cut effects. Those large impact, highly penetrant errors that cause the classic Mendelian disorders.
The big players.
Right. But consider the billions upon billions of much more subtle genetic variations, the common low-penetrance polymorphisms that we all carry in our genomes. These small differences may be conferring just marginal changes in enzyme efficiency or receptor binding affinity or inflammatory response. They're constantly interacting in the background, unseen.
They create a unique genetic landscape for each of us.
Exactly. They shape the underlying genetic architecture of the common, complex, multifactorial diseases that affect most of us eventually. Things like atherosclerosis, hypertension, type 2 diabetes, Alzheimer's disease.
They're not causing disease on their own, usually.
No, they represent silent layers of susceptibility, or maybe resilience. They often require a specific environmental factor, perhaps a high-fat diet interacting with lipid metabolism genes, or chronic stress interacting with neuroendocrine pathway genes, or a toxic exposure interacting with detoxification genes to unmask them and tip the balance towards a full-blown disease state.
So our genetics isn't just a predetermined fate.
Not at all. It's more like a highly complex, individualized landscape of potential risk and protection, constantly being shaped and reshaped by the choices we make in the world we live in. Something to think about. Keep diving deep.